We have one more student who will be presenting her work. And at the close of this, we will close our voting. So those of you who are watching, if you'd like to cast your votes, by the time Qidang is done, that would be great. So our next speaker is Qidang, and Qidang is at the University of Ottawa in Canada. And the title of her poster presentation is Bioinformatics Analysis of Adhesin-Binding Potential and ADME Tox Profile of anti-Helicobacter pylori peptides derived from wheat germ proteins. Chi, please go ahead.
Welcome, everybody. My name is Qidang, and I'm an undergraduate student at the University of Ottawa. And today I would like to present my research project titled Bioinformatics Analysis of Adhesin-Binding Potential and the ADME Tox Profile of anti-H. pylori peptides derived from wheat germ proteins. Okay, so firstly, H. pylori is an important human pathogen causing gastric infection and possible ulcers of over 50% of the world's population, and has been classified as class 1 carcinogen by the World Health Organization. And the infection is usually treated with antibiotic regimen. However, due to the emergence of multi-drug resistant bacteria, it is important to explore alternative treatments, and anti-adhesive therapy with bioactive peptides is considered one of the most promising strategies by interfering with the bacterial adhesion to the host cells. And food-derived bioactive peptides are gaining more interest as nutraceutical candidates, as they have been demonstrated to have therapeutic potentials. And there are also some research that prove that they are optimistic anti-adhesive agents. And so the objectives of my study were to validate in silico the anti-adhesive property of the previously identified wheat germ proteins, and then to evaluate the ADME tox profile based on the pharmacokinetics parameters. So starting with the initial database of 267 peptide sequences, we perform in silico analysis with digestion with pepsin, which is stomach enzyme at physiological pH to assess the biostability. And then we perform molecular docking to visualize the binding as well as to obtain the binding affinity of the peptides to the two most dominant H. pylori adhesins, BAP-A and SAP-A. And then we evaluate the predicted pharmacokinetics, physiochemical properties, as well as toxicity of the final 16 selected peptides. So the results here of the representative 3 peptides extract from the wheat germ proteins. And so you can see that there are multiple binding poses of the peptides to the adhesin SAP-A, as well as so they bind with medium to strong binding affinity. And the peptides were shown to be a poor oral bioavailability due to the high number of rotator bond as and the polar surface area. And so this could inspire more further research. As well, they have high solubility, and all are shown to be non-toxic. And they are also have low gastrointestinal absorption, which is desirable in this context, considering the targeted application of the peptides were to act as receptor analogues to prevent H. pylori infection. And so the bioavailability radar image shown over here of the peptides are quite similar to each other, and they're also similar in many parameters to the 3-sialylactose sodium salt, which is a compound derived from human and bovine milk that has been verified binding capability to SAP-A, as and also being involved in some clinical research in human and rhesus monkey. And so this suggests that the peptides could possess the same mechanism of action as well as biofunction in preventing H. pylori infection. And so in conclusion, the anti-adhesive property of the defatted wheat germ-derived bioactive peptides was validated, and based on the in silico ADME tox profiles, it is shown that they show the promising anti-adhesive activity. And this is my reference, and thank you for listening. I will be happy to take any questions.
Thank you, Chi. Very interesting talk and a very fun approach to this. So I have a question about your docking. So you carried out in silico digestion and you derived some peptides. I would expect that the digestion would take place in the stomach, right? Because it's a pepsin, is a stomach enzyme. But suppose there could be some carryover into the intestine. So when you did your visualization of your docking, did that happen at stomach pH or small intestine pH? Or would you expect it to not make any difference?
We perform docking with our regards of the pH. We only perform like the assessment of biostability with the pepsin-hydrolyzed pH. So that was the previous step to docking. And so that could be what we look into further, but what I predict that the low pH of the stomach, if it doesn't affect the peptide stability, but in the intestine, I think that the pH is a bit higher. So it could alternate the structure of the peptide. So we could also look further into that too.
Thank you. Do our judges have questions?
Yes, maybe I can start. So thank you, Chi, for this very nice presentation. Very interesting. When you speak about peptides, do you know the molecular weight of these peptides you were using?
Yes, so our molecular weight, just give me 1 minute, please. So our molecular weight are pretty high, so they're all higher than 500. So it kind of violates the Lipinski rule of 5. However, so they range in about maybe 800 to 1200. So it's pretty big, yeah.
Yeah. And my second question is about the purity of these peptides. Are you using really highly purified peptides, or it's just an extract containing maybe some side molecules?
Yes, so we extracted from, so we have like a hydrolysate from the wheat germs, and then we perform SDSPH as well, blotting, and then identify the peptides components by mass spectrometry. So of course that there could be some impurity involved, and so this could also affect the properties that we predict in silico. And so from the next step, we could validate in vitro, and then we could also look into the purity of the samples that we collected before too.
Okay, thank you.
Okay, I can go next. So Jidan, I think you did an excellent job. I wanted to know that you are actually an undergraduate student. Great job. Okay, my first question to you, where did you get these 267 peptides?
Yes, so this I did this based on the initial database. So there was a study before from one of the postdoc fellow in our lab. So she did so it was like in vitro experiment, and then they do like identification of the peptides based on the experiment. And so for the in silico work, that was the database that I worked on. So these are already verified with some properties against H. pylori infection.
Very good. Could you tell me how long did you spend on this project to date as you presented?
Yes, so because it's bioinformatics work, so it's a lot faster than if you do them with laboratory. So in terms of many that I use in these are web-based tools. So maybe like Streamline takes like a couple of weeks to like a few months. And then the docking part is the longest part, but in general, it is very fast to obtain result.
Okay, thank you. I know that's also need a lot of intelligence as well. Could I ask you what ADME ADME stands for?
Yes, so ADME is the pharmacokinetics parameters of absorption, metabolism, distribution, metabolism, and excretion.
Excellent, thank you.
Thank you.
Hello, Chi. Yeah, I agree with Dr. Wu's comments. As an undergraduate student, I think you did a wonderful job. Yeah, well done. I just have a quick question. Could you briefly explain when you do the in silico digestion analysis, why you only use the pepsin and didn't choose the enzyme from the intestine?
Yes, so the reason why we use pepsin is that H. pylori infection usually, because of the flow of the stomach, it's usually concentrated in the bottom of the stomach. And so in that area, pepsin is according to what I know, is the only protease available in that area. And so if there are any activity that we want the peptides to be, we would assess with the pepsin only.
Thank you.
Thank you.